Hello everyone, myself Dr. Parth Goswami, and today I am going to teach you about immune tolerance, that is a basis of autoimmune disease development. It's one of the most important topic from immunopathology chapter, and I will make this lecture very interesting. And after watching this lecture, you can surely understand the basic concepts of immune tolerance, and you can write down the long answer question, or you can attempt the MCQ in your exam as well. So it comes under the competency number PA 9.7, in which student need to define and describe pathogenesis of autoimmune disease. Immune tolerance is a basis for development of all autoimmune disease, right? So before we understand the pathogenesis of autoimmune disease, you need to understand what is immune tolerance. Because the most common mechanism of autoimmune disease development is breakage of tolerance. So what is immune tolerance? So, normally our body is not responsive to the self antigen, right? And so the autoimmune disease is prevented. If you remember my first lecture of immunity, then I have told you that, immune system is having the function of recognition of self from non-self. They can recognize the foreign particle, antigen, microbe, etc. So they will not responsive to the self antigen. They will tolerate our own antigen. That is known by the name immune tolerance, right? Immunological tolerance. It's a normal state of our body. So the autoimmune disease not develop because of immune tolerance. But somehow, due to any cause, if this immunological tolerance break, then immunity system will act against our own self antigen, and that is known by the name autoimmune disease. Rheumatoid arthritis, systemic lupus erythematosus, myasthenia gravis, all that are example of autoimmune disease in which the body produces the antibody against our own self antigen. All right. So which are the types of immune tolerance, right? We have seen that body is non-responsive to the self antigen, that is immune tolerance. But which are the types? So suppose if there is any self-reactive cell in our body, then how they remove? So basically there are two types of immunological tolerance. One is central tolerance, and the second one is peripheral tolerance, right? So what is the meaning of that? We know that there are two lymphoid organs, central and peripheral, right? The central peripheral organ consists of bone marrow and the thymus. So if the self-reactive lymphocyte removed, or they rendered inactive in a central organ of immune system, then it is known by the name central tolerance. But suppose they escape the central tolerance and reach into the peripheral circulation, and removal of self-reactive cell in a peripheral organ, that is known by the name peripheral tolerance. All right, so first let's begin with central tolerance. So it acts in a two way. One is central deletion, which is negative selection, and second one is central editing, which is a receptor editing. Very interesting mechanism. Let's see each one by one. So central tolerance means self-reactive lymphocyte removed in a central organ, that is in the bone marrow and thymus. All right. So first let's see what is central deletion. What is negative selection? So the name itself suggests here the self-reactive cell will be deleted, and that's why the name central deletion is given, right? So you know T cells and B cell get mature in the thymus and bone marrow respectively. But suppose they are self-reactive against the self antigen, then they will be killed by process of apoptosis. They will deleted. They will killed by the process of apoptosis, right? And that is under the regulation of autoimmune regulatory gene. AIRE gene will control this apoptosis process, right? So if you have the mutation in AIRE gene, then autoimmune disease can develop because the central deletion will fail. All right. Second mechanism is central editing or receptor editing. So this is very beautiful mechanism. See, suppose if the self-reactive lymphocyte not get killed by apoptosis, then central organ is having another mechanism, that is the editing of the receptor. Very beautiful mechanism. See, if the self-reactive cell not removed by apoptosis, then they will undergo different mechanism, that is receptor editing. Here the cell will rearrange the receptor antigen genes will be rearranged. So, there will be rearrangement of the genes so that there will be new receptor formation, right? So it will express a new receptor that is no longer self-reactive. That is known by the name central editing, right? Very interesting mechanism. So that these two are the central tolerance mechanism. All right. But interestingly, some self-reactive lymphocyte can escape this central tolerance. They will escape from thymus and bone marrow. right? So they will get removed by the process of peripheral tolerance. So now let's see the mechanism of peripheral immune tolerance. So there are four mechanisms of peripheral tolerance. One is energy, second one is regulatory T cell, third one is activation induced cell death, and fourth one is antigen sequestration. Let's see each mechanism one by one. First is energy. So in our T and B lymphocyte lecture, we have seen that for the full-fledged activation of the T lymphocyte and B lymphocyte, there is a need of induction of co-stimulatory activation as well, apart from MHC activation, right? Two signal is required for activation of T and B lymphocyte, and the second signal is co-stimulatory molecule activation. For the T cell, the co-stimulatory molecule is CD28, and for the B lymphocyte, co-stimulatory molecule is CD40, right? So suppose if there is an inhibition of such co-stimulatory activation, then we can render the self-reactive lymphocyte non as a non-self-reactive, right? That is known by the name energy. Energy means body will stop the co-stimulatory activation, and so the self-reactive lymphocyte can no more work, right? That is known by the name energy. In this mechanism, there will be inhibition of co-stimulatory signal. All right. Second mechanism is suppression by regulatory T cell. Now, we know very well that T lymphocyte is of three types: CD4 T helper, CD8 cytotoxic T lymphocyte, and third one is regulatory T lymphocyte. So, regulatory T lymphocyte will remove the self-reactive lymphocyte if present in our peripheral circulation, right? So, regulatory T lymphocyte express the two gene, that is CD25 and the FOXP3. CD25 and FOXP3, they will express these two genes, and that's why they will secrete interleukin 10 and transforming growth factor beta, which can render the self-reactive lymphocyte inactive, right? They will inhibit the self-reactive lymphocyte. So that is the mechanism by regulatory T cell. So if you have the mutation in the FOXP3 gene, then autoimmune disease can develop. All right. Third mechanism is activation induced cell death. See, suppose you have the self-reactive lymphocyte in your circulation, then they will activate two molecules. One is pro-apoptotic molecule, that is beam, bead, and bad. That is a stress sensor, and they will increase the expression of FAS ligand as well. Because of increased expression of beam, bead, and bad, that is a stress sensor, intrinsic pathway of apoptosis is activated, right? This beam, bead, and bad will activate intrinsic pathway of apoptosis, right? And by the expression of FAS ligand, there will be activation of extrinsic pathway of apoptosis. Understand? So, by activation of these two molecules, apoptosis is activated, right? So apoptosis is stimulated, and apoptosis will remove such self-reactive lymphocyte. But if you have the mutation in this FAS gene, or any of the gene that regulate this beam, bead, and bad, then apoptosis will not occur, and autoimmune disease can develop, right? All right. Fourth mechanism is antigen sequestration. Very interesting mechanism. See, there are certain immune privileged site in our body that is having the hidden antigen. For example, eye, testes, brain, all that sites is having the hidden antigen. Means they are having sequestrated antigen. They cannot recognize by our immunity. They are hidden. Why? Because they are not in contact with our blood or lymph fluid, right? So they are hidden. But whenever there is a trauma or the infection in this organ, eye, testis, or brain, if there is any injury, then because of, for example, post-traumatic orchitis, because of injury, this hidden antigen will be expressed and the immunity will work against it, right? So that can be the basis of development of autoimmune disease. Normally they are hidden, but because of injury or infection, they will expose. So the body can react with this hidden antigen, because body doesn't recognize it. They are the hidden one, but because of injury or infection, they are expressed. So body will react. So that is also the mechanism of autoimmune disease development, right? All right. So basically, This is the golden rule. There are three main principles of autoimmune disease development, or we can say three main pathogenesis or the mechanism of development of autoimmune disease. One is failure of tolerance. Which tolerance? Either central or peripheral. Genetic factors and the role of infection. These three factors responsible for autoimmune disease development, right? All right. So we have seen the tolerance. Now let's see the genetic factors involved in autoimmunity. So most of the autoimmune disease, runs in a family. And in our HLA lecture, in our MHC molecule lecture, we have discussed that, different HLA molecules related to the different autoimmune disease. For example, in the DRB1 is related to the development of rheumatoid arthritis. DRB1 related to the development of systemic lupus erythematosus, and HLA-B27 related to the development of ankylosing spondylitis. There are lots of examples. We have discussed many examples in HLA molecule lecture, right? So the genetic factor can be associated with autoimmune disease. PTPN22 gene is associated with type 1 diabetes and the rheumatoid arthritis. Most important MCQ. NOD2 gene is associated with the Crohn's disease development. Very important MCQ again. All right. Now, what is the role of infection in the autoimmune disease development? So, the infection can lead to autoimmune disease development in a two way. One is that, infection can upregulate a co-stimulatory molecule. So, there will be failure of tolerance, right? We have seen that in the tolerance, the co-stimulatory molecule is inhibited. So, self-reactive lymphocyte will work no more. But if there is an infection, then in the self-reactive cell lymphocyte, they will upregulate a co-stimulatory molecule, and so there will be failure of tolerance. All right. This is very interesting. Some virus or bacteria show the similar antigenic structure like that of our own host cell. That structure is similar to our cell, right? So the autoimmune disease develop because the structure is same, right? That is known by the name molecular mimicry. Because of the same antigenic structure, immunity cannot identify whether it's a foreign or self antigen. So in fighting with virus or bacteria, they will react against our own host cell as well, because the structure is similar, which is known by the name molecular mimicry. So the infection also sometimes having the role in development of autoimmune disease. All right. So that was all about the immune tolerance and the development of autoimmune disease. I hope you have enjoyed this particular tutorial prepared by me, right? I will be right back with a new video. Till then, take care and bye bye, right? Thank you very much. Yeah.